Welcome back, friends. I'm making this video to solve the problem that is going on. According to the video, is that I've seen the video just I've made and posted about this polymerase 2 mediated process of transcription in eukaryotic system. Is that the uh, drawings become hazier, so it's a camera problem, so it's a fault, technical fault. So in this, that's why I'm going to recapitulate what I have talked about the process of transcription utilizing the RNA polymerase 2, and then after that we'll be talking about the termination, which I've told you before that we will be making a video separately on termination. So before that we'll be going through this process in gist. Okay, so what will happen in case of this polymerase 2 mediated process? We again, three things are important. One is the promoter region of the DNA. Second thing is the polymerase itself. Third thing is the transcription factors. Now in case of polymerase 2 mediated process, we require both generalized transcription factors GTFs as well as the specialized transcription factors or STFs. Now in this case, the promoter sequence of this polymerase 2 mediated process must be divided into three different parts. One is the Tata box. From the Tata box, the downstream region of the Tata box, we are having the initiator box sequence, and upstream of the Tata box we are having the upstream control element, or UCE. We can write USE is there, so whatever UCE, USE, whatever you write, but the thing is it's an upstream element, or the region for the proper binding of transcription factors. Okay, then the very first step of this reaction in all like all the other reactions of type 1 and type C type is that in this case the binding of the generalized transcription factors, which indicates the place for the attachment of polymerase 2. So here the generalized transcription factors like TF2D, TF2A and TF2B will initiate the process. So at the very beginning, TF2D comes in and it will sit onto the Tata box region. Now this TF2D is a very important protein because it is having the Tata binding domain. The Tata binding domain of TF2D is having higher affinity towards the Tata region, or the TA sequence that is present in the DNA. Now it will come and it will bind with the Tata region with the help of the Tata binding region. We call it the Tata binding protein or Tata binding factor. You can call it like TBP or TBF accordingly. It will bind with it, and as it bind with this Tata region, that Tata binding factor region of this TF2D is made up with mostly beta strands. Now the beta strands of that is having polyphenylalanine residues. As it is having polyphenylalanine residues, it will kick these Tata regions in both the terminal ends and it will make the DNA bend -80 degree. Okay. Now after bending the DNA -80 degree, it makes hydrogen bonds, several 7 to 8 hydrogen bonds with this Tata sequence, and hold this bond, not actually 7 to 8, so forget this, small amount, fewer number of hydrogen bonds, and it will make this bended structure stabilized and combined with this Tata TF2D. So it is not only bending it but also holding the bended structure with itself. To further stabilizes the bend and conformation of the DNA, it will recreate TF2A factor. So TF2A factor will come and attach to TF2D along with the same region of Tata box, and it will further stabilizes the bended state of the DNA. After that, they will recruit TF2B protein. TF2B protein is the very first type of protein which is going to initiate and bind with this initiator box region. TF2B come and sit onto the initiator box region, attach to it, and then it will make a complex of DNA and protein. As it is making a complex of DNA and protein, it is the affinity towards this protein DNA complex for polymerase 2 getting increased, because polymerase 2 is having less affinity to naked DNA. It is having higher affinity to those DNAs which is bound with these factors. Now as now they are bind with the factors, this polymerase will have higher affinity towards this region. So polymerase must be recruited to this scene now. and the recruitment of the polymerase is conducted by another generalized transcription factors, TF2F. TF2F will hold the polymerase 2 and bring it to this reaction center to make, which is called the pre-transcription complex, which is just formed before the transcription process begins. After making this complex, this polymerase will again interact with the upstream control element to get a signal to move on, and this signal is provided by the interaction of RNA polymers with upstream control element via specific transcription factors or specialized transcription factors. So some specialized transcription factors along with their DNA binding domain bind with the upstream control element, at the same time bind with the polymerase via their activator domain or polymerase binding domain. It is also called the activator domain. It will activate the polymerase in this place. Okay, now after activating this polymerase must have the signal to move on, that it's the best place to your binding. You bind to the right place. Now it's your time to move on the forward direction. Here we mean forward means 5' to 3', the transcription. So for this signal to achieve for this polymerase, we must require a protein to provide it, and again the factor is TF2H. Now this TF2H factor will bind to this polymerase and it will signal twice. It will provide a signal to polymerase to be move on forward. It will also melt the DNA to create the path for this polymerase to move. Now how can it provide the signal to the polymerase? Polymerase is having the C-terminal domain, which is called the alpha CTD of RNA polymerase. Now this alpha CTD region of RNA polymerase is consisting of stretch of amino acid sequences. It is called 7 sequences. So tyrosine, threonine, serine, the sequences like that. Tyrosine, threonine, serine, like that. So there are a lot of sequences like that, 7 sequences. So this transcription factor 2H will phosphorylate these sequences. As a result of this phosphorylation of these amino acids that are present in the alpha CTD region of the RNA polymerase, the polymerase get the signal to move on the forward direction. And then this TF2H is also getting the energy from the ATP hydrolysis, and it will start melting the DNA strands from each other, open up the DNA to produce the open complex. In previous case, when everything is bound there, no melting of the DNA, it is called the closed complex. Now when it is get open, it is called the open complex. The open complex is formed, then nucleotides can come in, join it via the complementarity, then attachment of phosphodiester linkage, then elongation will be moving on. So this is the whole process of initiation of transcription and elongation in case of the polymerase 2 mediated transcription inside the eukaryotic system. Now there is control mechanism lies in different steps, because we need to control the situation, right? So we have inhibitors which will block the activity in some regions, and we are also having activators which is triggering the events. For example, we have seen lot of activators here. So let us take one example of an inhibitor, a protein called Tata binding protein. Remember Tata binding factor. It will bind here to bend the DNA from the Tata region. There is also a protein called Tata binding protein associated factor, or TAF. Let me write it here. It is called TBP associated factor, or TAF. This TBP associated factor, or TAF, is an inhibitory molecule which is going to control the binding of Tata binding protein, or TBP, to the Tata region or Tata box of the DNA. What it does, it is having a flap like structure. So utilizing this flap, this TAF protein will interfere between the TBP domain and the Tata box region of the DNA. So this is the Tata box, this is the TBP domain. It will produce a flap like structure. So let me draw this. It will be helpful for you if I draw. So let us just erase this. say here is our Tata box region. So this is our DNA and this is our Tata region. Here is our say Tata binding domain. Tata binding domain of this TF2D protein, and in between here we will have the flap structure of say Tata binding protein associated factor or TAF. It will block the binding of Tata binding domain with Tata box utilizing the flap. So if this protein, if TF2D will not able to bend the DNA, the whole process will be halted. Transcription won't be happen, because in those cases other factors won't bind, polymerase won't bind, and especially specialized transcription factors won't interact at the same time with upstream control element with polymerase. So it won't provide any signal to the polymerase. Even if the polymerase sit onto the place, it cannot move forward. So for the perfect transcription, we need the proper binding of this Tata binding protein with the Tata box region. But here it is tightly controlled utilizing the TAF protein. So when TAF is present, it will block the binding with this flap. Now when the flap is removed, when they need to go this transcription, when they need to carry out the transcription, this flap will be removed, and then it will allow the Tata binding protein to sit onto the Tata box. This is the signal that is coming out from the cell, that this is the time for transcription, this is the time for translation and all this. So this is a negative regulation that we can see. We have seen a lot of activity of activators in different regions, so I'm not going to talk about them. So this is the total system of polymerase 2 mediated transcription process. Now if we are focusing on the termination or the last stage of it, I just briefly discussed in my last hazy video about the termination process, but in this case let us consider it our topic and let us discuss about this termination video, termination process. Say okay, let me erase this first, full erasement. Yeah, so the termination process here is quite different from that of the termination of the prokaryotic system. Say in this case we are having the DNA, okay, and we are having the polymerase. So bubbles, so let me draw it like that, because bubble is formed, right, like that. Polymerase is there, say this is the polymerase pol2, and it is producing this mRNA. Now as it is going in this direction, this is say 3 prime 5 prime, so this is for the DNA, this is for the RNA. Okay, now RNA production is going on in this direction, holding this strand, this lower strand, as the template strand. Now what will happen, as the polymerase is moving on to this forward direction, it will encounter a signal. It will encounter a signal in the RNA. What is the signal? The signal is like that, A U A. More or less the signal is like that. So when it gets a signal like that, it eventually recruit some factors to this place. Some factors. And this part of our discussion is more likely to the polyadenylation process. So I'm not going to tell you the factor names and all this, but try to remember, try to understand the concept here. So it will get this signal. Suppose this is the stretch of the signal, A. So this is the signal, say, and further downstream it is having a signal CA. So when it gets signals like this CA, it will recruit some endonuclease. It is also called cleavage factor, CF. which is called the endonuclease. It is called cleavage factor or CF. Okay. There are also accessory factors like cleavage and polyadenylation specificity factor or CPSF, and also CST, cleavage specificity factor. So there is cleavage specificity factor, cleavage and polyadenylation specificity factor that will recognize these two regions. Okay, the proteins CPSF, cleavage and polyadenylation specificity factor, first recognize this termination signal. This is called the termination signal. This is called termination signal. Okay, so this termination signal, when they get this termination signal, they recruit CPSF at this point. This is CPSF, and also CST or cleavage stimulating factor in turn is going to bind with CA. This is CST. It is going to bind with this CA region or CA sequence. CST is going to bind with CA sequence. It is going to bind with this AAU or termination signal. After the binding of these special factors to these different sequences or specialized sequences, they will recruit the endonuclease, and the endonuclease here is a cleavage factor or CF. Now CF come to the place, and CF will cleave this sequence from this region. It will cleave just right after the CA. The CA is telling it cut the RNA from here. So it will bound with this whole structure, whole complex, cut it right after CA. And then what it does, it will provide us with this mRNA, where you are having 5 prime and 3 prime like that. But not only this cutting is enough, because remember, the process of polyadenylation, the process of 5 prime capping must be carried out simultaneously during the transcription. They are co-transcriptional events. So again, when we are providing and producing this whole chain, just right after few stretch of sequence production, 5 prime is modified, it is capped. So capping is done very earliest time, but after the synthesis of this mRNA, it is our duty to add polyadenylation to the 3 prime end of the RNA. But for the attachment of this polyadenylation, we require the polymerase enzyme. So polymerase will from here will attach polyadenine residues at the end of this newly grown mRNA. So from here on it cut it. So it is, now we are much more concerned about the termination. So whenever we get the signal, CPSF, CST will bind with it, then a cleavage factor will come and cut it from this region. After it cut this, right after the CA region, polymerase will start adding a polyadenine. But this is not the end of the process yet. Polymerase is not falling out just right after the event. It is going on and moving on in the forward direction. Right after some time, so when it is moving on in the forward direction, though the RNA which is required for us is produced, but still it is moving on in the forward direction. So it will produce a few stretch. It will produce a few stretch of, say this is the black colored thing, few stretch of RNA, small stretch of RNA. Then it will fall out, because after some time it won't find right kind of binding, right kind of affinity. It won't find it, and it will lose the interest from the DNA. But still, right after the synthesis of mRNA, that polymerase won't fall off just after that. It will carry on some time, and when at this time it will produce a few stretch of small stretch of RNA. This small stretch of RNA is worthless. So the fate of this RNA is degradation. Spontaneous degradation of this small stretch of RNA happens inside the nucleus. It won't be allowed to come into the cytoplasm. It will be degraded inside the nucleus, chopped out, and we take this mRNA. It is polyadenylated, and a protein called polyadenylation binding protein or PABP will come and sit onto this polyadenylation region, Sometimes it will hold both the ends, 5 prime and 3 prime. 5 prime is capped. It will hold these two regions and it will make a circular like structure. So it will make a circle like structure, say like that. So this is the capping region, say, and this is the polyadenylation region, and in the middle we are having this PABP protein sit onto and holding the structure together to make a circular RNA. This making the circular RNA is lies the importance of stabilizing it. So this RNA will be much more stable. It will not be vulnerable to the damage. Okay, so this is how it ends at the end, and I hope this will help you to understand the termination. No row dependence, no row independence, simple utilization of some factors again. So lot of proteins are involving a simple game. If it is played by a prokaryote, then it will involve small proteins, fewer amount of proteins. If it is played by eukaryotes, it will amount enormous number of proteins. That's the difference between prokaryotic system and a eukaryotic system. I hope this video will help you. Thank you.